All right, before we get into 16.2, let's reorient ourselves. In 16.2, we're going to be talking about antibodies. So we'll be working with stuff on the left-hand side of this diagram, the humoral system as it's called. Remember, antibodies are the proteins that are made by B cells that have converted into plasma cells, and they make lots of these antibodies. Okay, so we're going to talk first about the structure of antibodies and the broader types of antibodies that are out there. We will then talk about antibody response. So there's an initial primary response the first time you're exposed, and then a secondary one the second time you're exposed to an antigen. We'll talk about B cells and how they differentiate, and a little bit about the B cell receptor that's on there. And then we'll talk about how this all fits in and mention B memory cells here. Okay, so antibodies. They are a class of protein we call a glycoprotein. They're a mix of sugar and protein, and they give our adaptive immune system a lot of specificity. They're part of a broader family of proteins called immunoglobulins. We also have some key things, major histocompatibility complex, and B and T cell receptors that are in this family. We'll see each of these in turn. The antibodies, when they're made, they're going to circulate in the blood, in the lymph, looking for their specific target. When they find that target, they're going to bind to it and lead to the destruction of that target. There are a couple of different ways they can cause that destruction, as we'll see. So here's the general structure of an antibody. It is a series of 4 polypeptides that are all bonded together. So this has a quaternary structure if we go way back to the protein structure side of things. There is 2 kind of Y-like protrusions that come off here. On those, we will find the antigen-binding sites, and then there's kind of the tail here. This is called the FC, or the constant region. So we're going to look at antibodies, not in a huge detail, but I do want you to know that there are different classes of antibodies. They vary based on their constant region. So we have IgA, IgE, IgG, IgD, and IgM. We will talk about what each of these is a little bit. Really, the big one is IgG. This does most of the killing of microbes and pathogens. And of course, don't forget about IG-11. And for you real OG Star Wars nerds, IG-88 is the original IG. Okay, so I had to get my one joke in there. We got a lot of IGs in here. Again, we'll talk mainly about IgG. If we break this down into 2 parts, the bottom part, the FC, this is the constant region. That's where it can vary, AG, different types. But the top here, this is what we call the variable region. This is where things change, and this is what gives an antibody its specificity. So these regions, the antigen-binding regions, are different for each different type of antibody that recognizes each different antigen. So there could be thousands of different antibodies in your bloodstream right now, each of them having different variable regions that recognize different antigens. If we look at kind of the composition of what antibodies are out there normally, the main one is IgG. This is our main human immunoglobulin. It will move across the placenta. It binds to complement. It works in opsonization. But you will notice there are some of these, pretty much all of them, are not produced by the fetus except for IgM. So the mother actually transmits these IgGs across the placenta to help give developing fetuses some immune protection. The other ones, IgA and IgM, are the large 2 other groups. And D and E are very specialized. We're not going to talk about them too much. So IgG is the main one that does most of the killing of microbes. It is very simple in structure. It's just that Y-shaped, and it's the most abundant, as we saw. And it provides the majority of our antibody-based immunity. It works by that opsonization, where it attaches to pathogens and helps phagocytosis occur. It can also do something we call neutralization, particularly of viruses, where it binds to the surface and blocks their entry. It can activate that complement system we talked about, and it is the only one, as I mentioned, that can cross the placenta to help provide immunity to the developing fetus. So IgG is really the big one that we should know about. Here is a representation of how it actually goes about destroying microbes. So one way is neutralization, like I mentioned. So here's a virus, and on the surface are all these spike proteins. These spike proteins are critical for tricking the cell and getting into the cell to replicate. IgG antibodies will come along and recognize and bind to these spike proteins, blocking them, essentially gumming up the viral particle, neutralizing its ability to get into the cell. If a virus can't get into a cell, can't replicate, it's out of the game. The other way that this can work is binding to the surface and then leading to opsonization, which is a special type of phagocytosis, where the phagocyte, like a macrophage or a neutrophil, grabs onto the little handle of the antibody and uses that to help pull the microbe into the cell, and there it gets degraded. So in both of these examples, you would notice this is the host cell down here. Antibodies only work outside of the cell. So as we'll see, if something gets inside the cell, there is a different response that we'll talk about in the cellular side of the adaptive immune system. The other antibody, isotypes as they're called, IgA. This is secreted across mucosal surfaces, so it actually is secreted in tears. It can be secreted in breast milk, so it can be transmitted to newborns through that, and it's actually in saliva as well. IgM is an interesting one. It can form like a huge wheel shape. I'll show you that in a moment. But it can also be monomers on B cells, and it helps form the B cell receptor. IgM is not actually super specific to the antigen that it binds to. It doesn't recognize it super well, but it is the first one that is produced in response to an antigen. So we will see that IgM gets made, but then we switch over to IgG later, which gives us more specificity and better ability to destroy pathogens. D can be found on the surface of B cells, helps them differentiate, and E is on the surface of mast cells. This kind of helps amplify the body's response. Those aren't super critical. IgG, IgA, and IgG and IgM are the big ones, actually, that we're going to talk about. So IgA actually forms what we call a dimer. It's two of the Y-shaped molecules stuck together. And IgM can form this giant pentamer, so 5 units of all these Ys that form this giant wheel. So the different structures here. Remember, IgG was just the single Y shape. Okay, so let's talk about what happens when you encounter an antigen and how we respond to that. Okay, so an antigen comes in, you're infected with a microbe. That is going to find and bind to a B cell receptor that is specific to that specific antigen. We'll talk about this in a sec. When that happens, the B cell will be activated. This is going to begin a process called proliferation, which means it's going to start dividing it and making copies of itself, and differentiation. The B cell is going to be bound by the antigen and differentiate into what we call a plasma cell. These plasma cells have lots of endoplasmic reticulum. That is where proteins are made, so they're basically little factories for producing tons of antibodies. Additionally, B memory cells will be created. They differentiate into B memory cells. So let's look at a graph of serum antibodies versus time. In blue here, we have IgM. You will see that it is the first to respond, but it takes a long time. We don't get peak until after a week. So we have our primary infection, and then like several days later, we get peak IgM. But IgM is not very efficient at targeting the microbe. So we switch at this point over to making IgG, which is here in orange. And that bumps up and will neutralize the pathogen. So that then will drop off. Okay, so that's after our first response. Where is this happening? Well, as we mentioned, the antigen-presenting cells are bringing these to the lymph nodes. Also, free-floating antigens. They're coming up here and binding to B cells, which differentiate into plasma cells or memory cells. Those are going to travel through the bloodstream to the bone marrow, where they're going to hang out, and the plasma cells are just going to pump out antibodies that will circulate in the blood and go back to the site of infection. Okay, that's the first response. Those memory cells are key, though, because the second time we encounter the pathogen, or if we're talking about vaccination, when we get a booster dose, something interesting happens. Right here, it took at least a week to switch over to IgG. When we encounter something the second time, right here, you will notice that almost instantly, within a day, we start producing tons of IgG. That is because we already have our memory cells produced. They're waiting in the bone marrow. They stay there for a long, long time, years even, decades, waiting to encounter that antigen again. And when they do, they're ready. They're primed, ready to make that IgG. So that is why the second time you encounter a pathogen, usually you don't get sick from it because you can neutralize it much faster. This works the same way, and you will notice we have a lasting IgG production here. So this works the same way for vaccination. We give you an initial dose of the vaccine, and you make memory cells to it. Then you either get a booster dose like we did with the COVID mRNA vaccine, and that causes a huge spike in IgG production, or you've got your initial vaccination, you have memory to it, and you encounter the actual pathogen. You probably won't get sick from it, or you'll be sick for much less because you can respond to it much quicker. The great thing about vaccination and boosters is that we don't have to actually get infected with the pathogen. Remember, when the pathogen infects you, it can cause all kinds of damage to your cells and to your body. So the vaccination does not do any of that damage. You might feel some inflammation, some symptoms of that, but it's not actually damaging your body like the pathogen would. So the symptoms of immunization are due to the immune response, not because you're actually getting infected with anything. So I want to reiterate, these B memory cells are key to why we can mount the response much faster the second or subsequent times. This protection will wane over time, right? It also is only still recognizing that specific antigen. If the pathogen mutates, like we've seen with COVID, your response will not recognize necessarily the new antigen that is produced. So, immunization is awesome. We don't have to get the disease, no damage to our cells. Yes, you might feel some symptoms due to the immunization, but that is because your immune system is responding to the immunization and producing a response that will then protect you if you ever encounter the pathogen. So how does this work? Well, the B cells rely on a special type of receptor called the B cell receptor. It functions in two ways. There is a mechanism of activating B cells that does not need T cells. We call this T cell independent. This only works for very specific types of antigens, like carbohydrates, that have a very repetitive structure. There'll be free-floating antigens floating around, and if they're very repetitive, they will bind to this B cell receptor on B cells. It looks like an antibody because it's very closely related to an antibody, and it will bind a specific epitope. And this process will activate the B cell, but this is the T cell independent form. The other type is T cell dependent. So in this example, the B cell receptor, it binds to antigens that don't repeat very well, but it requires activation by a helper T cell. So this helper T cell, it's going to have got an antigen from an antigen presenting cell, and it will come along. And so here's the antigen presenting cell. It gives it to the helper T cell. That takes it over to a B cell and activates that B cell. That will then go off and make plasma cells and memory cells and start producing large amounts of antibody. We will come back to this in 16.4 when we talk about the full process. What I want to talk about right now is how B cells make specific antibodies. Each B cell only recognizes one antigen. That seems crazy, but it is how the system works. So here we have a diagram. We have 4 different B cells. Each of them has a different antigen binding domain here. So it has, this one will recognize red antigen. This one recognizes yellow, this one blue, and this one green. Okay, all of these are made, and there's hundreds, thousands of these that get made. They wait and hope that they will be chosen, I guess is the best way to put it. Along comes the green antigen. It binds to cell 4. Okay, cell 4 has now recognized its antigen. It goes through a process called clonal expansion. It starts dividing. It makes copies of itself. It says, hey, I recognize my antigen. That's probably because there's a problem. I got to make lots of copies of myself. It also makes memory cells and plasma cells that make the antibody that recognizes the green antigen. Okay, this sounds a little complicated and it's a little overwhelming, but let's watch the animation. Hopefully it'll make a little more sense.
Each B cell circulating throughout the body or harbored in a lymphoid organ is programmed to synthesize antibody that reacts with a single epitope. In a process called clonal selection, the antigen selects which B cell clone proliferates and differentiates into antibody-producing plasma cells or memory B cells. B cells display their antibodies on their cell surfaces. Antigen binding activates a naive B cell, a B cell that has not previously encountered antigen, leading to its proliferation, or clonal expansion. A single B cell can proliferate into many offspring. Some of these offspring differentiate into plasma cells. Plasma cells produce and secrete large amounts of antibodies specific to the original activating antigen. Others differentiate into memory B cells. Memory B cells do not secrete antibodies themselves, but are primed to quickly become antibody-secreting plasma cells if the host is reinfected with the same pathogen in the future. These photos show a resting B cell, before activation and proliferation, and a plasma cell that is producing antibodies. Notice the increased size of the plasma cell compared with the B cell. The plasma cell cytoplasm is stuffed with endoplasmic reticulum needed to support the incredible increase in protein synthesis associated with antibody production.
All right, so this clonal selection happens every time we encounter an antigen. So copies of different cells get made for different antigens. But that leads to a key question. How can we have so much variation in the B cell receptors? How can they recognize so many different things? There's hundreds of thousands of antigens out there. And there has to be a way to make these different proteins. but they can't be coded in the genome. That would mean you would have to have hundreds of thousands of different genes just for these things. So this is where things get weird. We've talked about the genome as if it stays the same, right? Maybe occasionally there's a mutation or something. In B cells, weird things happen in the genome. There are an estimated 10 to the 11th, so that's 1 with 11 zeros, different types of antibodies that we can make. However, each person only has 1,000 genes or little gene segments that are involved in forming different antibodies. So how can we get 1 with 11 zeros from 1,000 different gene types? Well, this is where it gets weird, like I said. Antibody genes can actually rearrange themselves in the genome of a differentiating B cell. So here we have the DNA of a normal cell, okay? In there are coded all these different parts for making different types of antibodies. When this cell differentiates into a B cell, it picks different parts to put together. So V2, it picks that, and D4 and J3, it puts all those together. And then it's got all the different heavy chain bits. This is M, D, G, right? All the different antibody types, the isotypes. And when it switches isotype, it actually cuts out the extra bits. So this is going to make IgA here. If we had this one with gamma, it would be IgG. So this is kind of mind blown, right? Like, weird things are happening here. But through this process of rearrangement of antibody gene segments and a higher random mutation rate, and some other clever tricks, we can make unique B cells that can recognize all these different antigens. Okay, so that is a lot to take in, I know. Let's focus on antibodies. They're Y-shaped molecules. They have their 2 heavy chains, 2 light chains. We have those 5 different classes or isotypes: IgA, M, G, D, and E. Each of the antibodies has those kind of 2 arms of the Y shape that recognize a specific antigen. And these are made by really variable ends, as we saw. The tail part can be specific to certain receptors and used in opsonization. When we have an antibody response, the first response starts with B cells differentiating into plasma cells and memory cells. The plasma cells make lots of antibody. The memories go to the bone marrow, stay there, keep that memory alive. We start with IgM being made, but it's not as strong at destroying the microbes. So later in the response, we switch over to IgG, which is much better at targeting microbes. Then we keep that memory around. So the second time we encounter an antigen, our response is much faster. We make IgG quite rapidly, and usually that helps keep us from getting sick with that pathogen again. This all relies on the B cell receptors, right, to get that antigen. And then they go through clonal selection. Once they bind their antigen, they start making copies of themselves and divide out. And to make this diversity, we have 1,000 different genes in our genome. They kind of mix and match each other to go into a B cell to make all these different possible antibody combinations. Okay, that's it for 16.2.